biologist and involved in investigating genetic encoding. As you will see from his accent, although he works in the States, he actually is British, and he studied at Oxford and at York and here in Cambridge, and then moved to the States, where he has been working at the University of Maryland in Baltimore Country for a significant part of his time there. He also had a stint in Hawaii and one in Princeton, I believe. So Hawaii one sounds like a really possibility to those of us at the end of a British winter. But anyway, we're really looking forward to hearing you talk, because you bring your biology and also faith insights into it deliberately, don't you? So over to you, Stephen.
Well, thank you ever so much. Thank you. Let me stand up so you can see. You'll notice there are cultures at work within academia, and biologists are famous for being PowerPoint junkies. We live off our slides. I do live off them. They're really my flashcards. They're my cue cards to remember what I'm trying to say to you. But if anybody wants to see them and suddenly realise they can't at the back, there are spaces at the front. Don't feel awkward. That just occurred to me as you were talking then. And I'll stand up for the sake of trying to be visible and trying to let you see me. Oh. Having said that,
it's probably just as well we didn't record all that nonsense. So thank you very much. This is not quite my first time talking to Faraday, but every time I am invited, yes, my faith life is very real. I'm a Christian in science who finds the two constantly co-evolving. But I'm never quite sure that I'm hitting the right note for Faraday. I never quite know who I'm talking to. So, apologies to Ruth, I've slightly modified my title. And I want to make it clear from the get-go that I would like you to be able to help me steer this presentation, particularly Q&A, but even the presentation, towards what it is you need to be hearing to make your weekend useful. I've chosen to change my title slightly to say Faith Insights from the Central Dogma of Molecular Biology. And that will all be unpacked as we move forwards. But I had to remind myself what Ruth's mandate was to us as speakers. And here's my synopsis. She said that important questions for humankind and for Christian theology include: are the processes of the living world ordered or chaotic? Is evolution compatible with purpose? Is there more kindness than cruelty in the world? And how unique is our genetic code? That last one is me, and I feel a bit shamefaced about it because it sticks out like a sore thumb. I mean, we've got three questions here that are broad and deep, and then one that's extremely specific and refers to a piece of biology that I happened to be doing actually mainly when I was here at Cambridge at PhD back in the 90s. So I've chosen to change my title to really try to ensure what I know was Ruth's aim, which is that in telling you about work that I've done with the genetic code, I am addressing the questions: are the processes of the living world ordered or chaotic? And is evolution compatible with purpose? For my money, this number 3, is there more kindness than cruelty in the world, is perhaps the paramount question that comes out of evolutionary biology. And before I've finished, I want to make it absolutely clear that a comfortable theistic evolution model in which God, to paraphrase Jeff from last night, God creates the processes that then produce sentience. That's good in as far as it goes, but it leaves some mighty big questions about suffering, because the only way in which evolution proceeds is through death and suffering, the way we understand it. Now, that is not my domain, and I'm so glad we have theologians here to take over. But hopefully, before I've finished, I'll say something about these two and really cause you to be asking questions about this one. That's my aim for the talk. Also, I'm going to begin and end with this slide. Ruth makes it very plain that along with the rest of Faraday, our job here is not to be waging an endless, stale debate between evolution versus anti-evolution. So I'm quoting here Intelligent Design, and I'm quoting their manifesto, known as the Wedge Strategy, not because I'm here to debate intelligent design, but because I think that at their best, when I have found myself trying to address intelligent design, it causes me to think deeper and harder about questions that, as a believer in evolutionary research, we need to be thinking about. Here's a quote from the manifesto of the intelligent design community. The proposition that human beings are created in the image of God is one of the bedrock principles upon which Western civilization was built. Yet, a little over a century ago, this cardinal idea came under wholesale attack by intellectuals drawing upon the discoveries of modern science. Thinkers such as Charles Darwin portrayed humans not as moral and spiritual beings, but as animals or machines who inhabited a universe ruled by purely impersonal forces. The cultural consequences of this triumph of materialism were devastating. And it's really that last sentence, not the bit that I put in bold. But the point of the intelligent design community and the point of most anti-evolutionary work is that there is a perception among them that the cultural consequences of scientific insights have been devastating. If you go to the Creation Science Museum in America, by the time you've finished your tour, you will end up in a simulated inner city, deteriorated inner city environment, looking at graffiti and drug addiction, and learning that that is the outcome of particularly evolution, but of the scientific revolution in general. So that's the sort of the meta-awareness that I would like to put into you before I begin. That's where I will finish, and that is where I think it is worth us spending some of the time talking, although it's your time, so you tell me what you want to ask questions about. Oh, I forgot to jump this slide in the first beginning. I would take issue with several things here, right? But one of them is to say that this didn't begin with Darwin and it didn't begin a century ago. And I think many people in this room would know better and would trace it back to the Enlightenment in the 1700s, perhaps even back to the Renaissance. And the concept of a machine-like universe really is one that we could trace back to, say, Copernicus. And then watching through a series of scientific investigators who uncovered a very different model of the solar system from the prevailing model that the Middle Ages had inherited all the way from Greco-Roman times. The Greco-Roman model through to the Middle Ages, geocentric, Earth at the middle. We're literally the middle of everything. And our species is the middle of everything. And then through the insights of these astronomers and physicists, or as they would have called themselves, natural philosophers, we place the sun at the center. We learn that the orbits of the planets are no longer an expression of God's motivating energy that's revolving in visible crystalline spheres, but rather a result of gravity. And by the 17th century, if you're a rich, well-to-do gentleman, you might well own an orrery which celebrates this series of findings by showing you a mechanical universe in which if you turn the handle, everything obeys the laws of physics and perfectly describes the way that the planets move around the sun. That really is the machine-like universe. Darwin comes at the very end of that tradition, and to be brutally honest, he was a romantic, not an Enlightenment thinker. He was a geologist, not a naturalist. A lot of misapprehensions about who and what he was. But I would like you to think about what we talk about today and in this talk in a larger context of creating an impersonal, machine-like universe. Where does that leave us? You know, because Ruth invited me to open up, to be here talking to you, that I'm with her. I don't think there is any, I think there is more awe, wonder, grace, the more we discover. But clearly learning to articulate that and learning to communicate that, those of you who are teachers, theologians, ministers, that's our challenge, and we're hopefully here to reinforce one another in that message. Last thing, I'm sorry for the long preamble, but there's a lot I wanted to say before talking about the genetic code. This is stolen shamelessly from one of my other lectures that I would give, that is much more about the nature of truth. And I just want to state this as a personal statement that I hope guides not only this talk, but the rest of the weekend. And it boils down to this Venn diagram, okay? I stand here convinced that the truth that I can understand is a tiny subset of truth. To me, that's my interpretation of postmodernism. And therefore, I present what I think I understand, what I think I'm learning through the work that I do, very really hoping that you're going to come back at me in the Q&A time, showing me new things. That's to me the point of presenting. And I hope that will be a characteristic of not only this talk, but the rest of the weekend. We know that science is incomplete. We know that logic is incomplete. I believe my understanding of faith is incomplete. I believe absolute truth rests with God. And the joy of an occasion like this is to bring together so many different ways of knowing that we can draw inspiration and knowledge from each other. Okay, so part one, what is the central dogma of molecular biology? Can I just ask, quick show of hands, who knows? Have I said central dogma? Good. Okay, so sorry, but we're just going to spend, because you couldn't get more important, more fundamental to the biological revolution than to understand the phrase the central dogma of molecular biology. A couple of times in the talk where I'll just pause to ask a question. This one is really a brain teaser in many ways, but if I were to show you this sequence of dots and dashes and ask you, is that Morse code? Any instant reaction?
No,
Why not?
there's no gaps in it.
Thank you. Okay, that's a good start. Anything else? I'll leave that with you for a bit. Okay, the reason I'll leave that with you is I want to talk about the genetic code. And the way I want to begin is with something startlingly obvious. Clearly, the living world is beautiful and it is extremely diverse. David Attenborough would not have had the glorious career he's had were this not true. Right? The next point is that as a bona fide biologist, that does not represent diversity of the biological world. That represents one tiny, tiny, tiny sliver of the living world. That would be an objective measurement of life's diversity, measured in terms of differences in DNA, in genetic sequences. Everything has genes. Actually, everything has a lot of stuff in common, right from bacteria to oak trees to you. And if you build a evolutionary tree based on counting the differences in sequences, implying that those are mutations that have occurred over time, that's what gives us our evolutionary trees these days. We no longer do them from morphology, we do them from differences in DNA sequence. This is the tree results, and the point really is that fungi, animals, plants, everything on that previous slide is the tippy tip of one twig. Okay? Now, to you and I, subjectively, anthropocentrically, that looks like the diversity of life, because it's all about our size and it's glorious, and David Attenborough makes beautiful TV shows about it. But from an objective point of view, most of life is invisible to the naked eye, and looks the same if we stared at it under a microscope. There are three domains of life. The eukaryotes, the bacteria, and the archaebacteria. The archaebacteria are as different from bacteria as they are from you and I, but under a microscope they look identical to bacteria. We didn't even know they existed until the molecular tools developed for us to be able to count changes in DNA and make a more objective assessment. So that would be a true measure of life's diversity, far in excess of what that previous slide showed you. And yet the truly remarkable thing is that if you scratch the surface on that diversity, everything is the same. At the molecular level, everything is doing the same thing. My work tends more and more towards the origin of life. So I say it half-jokingly, but more and more seriously, particularly when I'm listening to Jeff last night. I can't tell the difference between an oak tree and a human being. I don't know what that is. I don't know any developmental biology. I learned more last night than I have done in 10 years. I really thank you for that. Because from my side, this is the sort of fundamental that is universal, true, and has been going on for a very long time. This, to cut a long story short, is the central dogma of molecular biology. And we'll go through it piece by piece by piece, but the cartoons that you're seeing are relatively atomically realistic representations of the molecules involved in life. And the central dogma in a phrase says DNA makes RNA makes protein. DNA makes RNA makes protein. These days it's fairly common knowledge that genetic material is stored in DNA, and we were hearing about that yesterday. We had a lot about proteins yesterday, and if you're not familiar with molecular biology, I guess the next thing I can tell you, I'm going to zip forwards. Yeah, horrendous slide there. But I'm going to zip forwards and tell you that these things called proteins are three-dimensional shapes that each catalyze a specific biochemical reaction by virtue of their shape. They contain the right cavities, the right surfaces, to bring things together that then react. So each protein typically catalyzes one biochemical reaction. That's the job of proteins primarily. Put enough of those things together, and you have a network of reactions that are life. That's the business end of life. Your cells right now are a bunch of proteins catalyzing a bunch of chemical reactions. So the middle bit is simply a statement then that, actually we'll stick with that one for now, that this is the inert genetic information, blueprints with all of the provisos that we learned yesterday, and thank you for that. I think you actually made a very powerful point to me because I'm more of a metabolism guy. And I'll say that this is information and it is sort of blueprinty in some senses, but it is totally inert. It first gets little lengths of that get transcribed into a chemical sister language known as RNA, atomically very, 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 very similar to DNA, but it's just like a temporary write form. It's as if this were in the file cabinets, and in order to take it to an office where it's acted upon, you open the file cabinet, pull out one sheet of paper and copy it onto something more transitory. That's the RNA. You then take that piece of paper down to the office where you're going to do something with it, and that's where it becomes a protein. It's translated into a different chemical language of proteins. Proteins are built as linear sequences, but they have the curious property that when you build a linear sequence, they will spontaneously scrunch themselves up into a three-dimensional shape, a unique and reliable three-dimensional shape. And it's that shape then that carries the functionality to catalyze a biochemical reaction. Okay? Several things worth mentioning here. One is that all of this is a series of linear polymers. We can use a linguistic metaphor, in fact we do. And as molecular biologists, we're so ingrained to use a linguistic metaphor, we forget to think how interesting that is. But everything is written in linear sequences. DNA is a linear sequence of letters, just like the ones you see whizzing past on the Jurassic Park computers when you watch the movie, the famous GC, A, and T. That's a linear sequence, one very, very long sequence. Each of your chromosomes inside your cells, just linear, has a beginning and it has an end. And there's just 4 letters involved. RNA, as I say, very, very, very similar chemically to DNA. So a linear sequence, 4 letters, spelled out the same way. And then proteins are built as linear sequences. They're built from a very different chemical alphabet, and more about that in a second. This time there are 20 letters that you can use. But they are built, once again, as a linear sequence, just like a word. But they're a word that has this peculiar property, that you spell it out like a long German piled up compound noun, and now it just scrunches together again. And so by the time you're finished, you've got the three-dimensional shape that now takes its reliable place within metabolism, catalyzing a bunch of chemical reactions, which include but are not limited to synthesizing the DNA, making a copy. So there's a circularity to the whole process. We tend to always write it linearly, left to right. DNA makes RNA makes protein. And sometimes we forget, unless we listen to a good talk by Jeff, that the whole point of proteins is that they come back again and make the start of it. So really it should be drawn as a circle. Okay. So, genes and proteins are written in fundamentally different chemical languages. There are 4 letters to the genetic language, whether it's DNA or RNA. They're shown at the top there. And there are 20 letters in the protein language. Those 20 letters are actually known as amino acids. So when you're buying from, the vitamin shop in Britain, your amino acids, you're just buying one of the 20 building blocks that goes into making a protein. Point being that those languages have nothing chemically to do with one another. No natural affinity, no natural relationship, they're just different. Okay? So the real heart of this whole process is a process of translation. Not just metaphorically, but literally. You've got 2 biochemical languages that one needs to be translated into the other one. So the genetic language is read and translated into the protein language by a curious molecule known as tRNA, which is horribly inaccurately shown there as a grey blob. And as long as there's no biochemists, I'll get away with it, sorry. I know a couple of you are wincing at seeing this. But its point is that it really has there's nothing intrinsically useful to know about its detailed structure, other than at one end, it recognizes and reads a specific code word of genetic material, length 3, and at the other end, it carries one of the 20 amino acids. And you have a suite of these things, each of which recognize one or more code words and carries one specific amino acid. So given the right set of these adapter molecules, life is able to translate genetic language into protein language. That's the real trick to life. It's not at all clear how or whether or why amino acids would interact with nucleic acids were it not for this arbitrary translation molecule. And when we talk about the genetic code, all we're referring to is the set of these molecules that is at work in a given cell, which can be represented on paper by a simple table that says here are all the genetic code words that are possible, written out in blue, and each of them corresponds to one, and exactly one, amino acid meaning, because there is a tRNA at work. So the genetic code is a lookup table that you will find usually on the inside cover of every textbook of biochemistry, cell biology, genetics, and so on, even introductory biology. And over time, students will tend to get a feeling until they, without actively memorizing it, we tend to know these days that GGG will always be translated as glycine, for example. Because it's very central, it's happening everywhere, it's one of the most fundamental features of life as we know it. The genetic code is the rule book for the translation process. And this is a way in which it is eerily similar to the Morse code. My own personal answer to why that wasn't Morse code is to say that dots and dashes don't mean anything unless I've given you a rule book. Likewise, as many of us biologists, we get frustrated with an increasing misuse by the scientific media to refer to DNA as the genetic code. Doesn't mean anything. Genetic code does not exist unless you have a rule book. That's what we mean by the genetic code. In fact, we know that this code can vary slightly in some lineages, which of course completely changes the meaning of DNA. There's no inherent meaning to DNA. As I work in the interdisciplinary studies department now, I'm fascinated talking to folk who think about translation in all its contexts, such as biblical translation. But to recognize that meaning is often implicit in the rules that aren't mentioned, and that the work of a translator is to carry into the translation concepts, baggage about meaning. And that's the only way you're ever going to extract meaning from a text. That's what we all do. I mean, that's what a linguistic philosopher will tell you we do when we read a book, right? Think about that childhood name of the kid who really annoyed you at school, and every time you meet somebody with that name, you have a slight negative reaction. That's translation, right? So there's Morse code. Life is doing something eerily similar in a number of ways. Notice that in Morse code, the dots and dashes is a language, a binary language, just 2 letters. Whereas it's being translated into the English language of 26 letters plus some basic punctuation. Well, the genetic code uses 4 letters in DNA, but it translates into 21 meanings, 20 amino acids, and a single piece of punctuation that means stop translating here. Very, very similar. Those of you who are older computer scientists will remember this for ASCII. Again, very similar. There is a series of ones and zeros that corresponds to every possible character that we would use in an email. The computer just uses ones and zeros, we see English, there's a translation process at work, the genetic code there is known as the ASCII table. Over and over again, humanity in its ingenuity has invented things that biology apparently invented billions of years previously. So, that's the central dogma of molecular biology. It's actually called the central dogma of molecular biology. We say that with a straight face every time. Thanks to Francis Crick, another ex-Cambridgian, who named it as such in the late 1960s as this was being discovered. There were at least 4 Nobel Prizes I can point to that were concerned with the things I've just told you. That's how important it is. Crick, of course, got a Nobel Prize for figuring out the structure of DNA, along with Watson. We can certainly point to another Nobel Prize that used very clever, elegant radioactive labeling to determine the difference between genes and proteins. Another Nobel Prize in the early 1970s for understanding that proteins, when they're built as a linear sequence, carry enough information to fold themselves up into a 3-dimensional shape. And of course, the Nobel Prize just for discovering the genetic code. So 4 Nobel Prizes within a decade just for uncovering this, because this revolutionized biology. This is responsible for everything that's happened since in biology, such as genetic engineering, building evolutionary trees, establishing evolutionary relationships. The reason that we're dealing with a world of GMOs is that this is so universal that once we'd understand it, it was only a matter of time before the technology came along for us to take a gene out of a fish that lives in Antarctic ice waters and put it into tomatoes, so that the tomatoes don't go squishy when you refrigerate them. Because they're all doing the same thing, so if you can find a gene that keeps Antarctic fish happy, you can take it out, snip it out, plug it into a tomato, and it works just fine. Right? Scary, but fine. Because everything is doing the same stuff. One final anecdote about this. Crick later admitted in an interview he had no idea what the word dogma meant. He was a committed atheist, so he wasn't very familiar with words like dogma. He believed it meant central truth. So he actually named this as the central truth of molecular biology. I think it carries endless irony because, like any good dogma, it carries us a long way, but it's full of assumptions and oversimplifications, which we'll spend the next 5 decades unraveling. But there you go. Okay. So it's been in existence for 3.5 billion years, at least, and counting. We believe that our planet is 4.5 thousand million years old, 4.5 billion years old, and we believe that this system had evolved by at least 3.5 billion years ago. It's been unchanged for 3.5 billion years. Life, evolution, has failed to upgrade it, improve upon it. It works that well. That's why it's universal. That's why it's at work in everything from bacteria to human beings. Something kind of important to bear in mind about it. It uses just 6 types of chemical elements. Those 4 letters of DNA or of RNA, and those 20 amino acids, all just use 6 chemical elements. Carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Think of that grand periodic table. And probably the best excuse we biologists have for not knowing enough chemistry is we don't have to really get beyond the first row of the period. It's fine. It's all okay. More about that in a second. But it's remarkably simple. It's remarkably long-serving. It is 3.5 billion years, and not one living system has found the need to improve upon that. And 21st century biology, as our tools get more sophisticated, we're realizing that life is far more widespread on our own planet than we ever dreamed. That's sort of the poster child of a class of organisms known as extremophiles. Extremophiles because they live in conditions that to us seem extreme, such as boiling water, or pHs that are equivalent to battery acid, or to the bleach that you'd use in a household sink. Organisms that thrive at minus 20 degrees centigrade. Organisms that live at 100 atmospheres pressure, deep below the surface of the Earth. Now we're learning to probe for them, they're everywhere. This guy is just awesome because he can do it all. He's called Deinococcus radiodurans. He's got the nickname of Conan the Bacteria. Nothing can beat this guy, right? So Deinococcus can survive cold, dehydration, vacuum, acid, the highest doses of radiation of any other living organism, and so on. We're listed as the world's toughest bacterium. But no matter where we look, we're finding life now. No matter where we look on this planet, we're finding life. In fact, we can reliably map organisms that we know of on the surface of this planet to environments elsewhere in the solar system. NASA is increasingly cautious about the extent to which we may have inoculated Mars. We didn't realize it, but we were carrying bacteria that are thoroughly viable when they arrived. Right? Interesting thought. And all of that is using the central dogma. All of that is using the same language of 4 genetic letters, 20 amino acids, 6 chemical elements total. None of it has found a need to upgrade that system, to make it more complex, to introduce new chemicals. None of it. Okay. So, I think the reason that Ruth invited me to talk about the genetic code was the PhD work that I was doing in Cambridge almost exactly 20 years ago. Please don't think this is cutting edge. I'll try and be a bit more up to date before we finish. But we were using computers to study properties of the genetic code here in Cambridge in the late 1990s. We were really picking up on a very old idea from way back in the heyday of discovering all this stuff, when a guy called Carl Woese noticed that amino acid meanings are assigned to genetic code words in a non-random pattern. Here are color-coded the amino acid meanings according to a specific property of the amino acids. This property, loosely speaking, is hydrophobicity, which is the extent to which the amino acid likes water or repels water. Think of oil and water when you're boiling some spaghetti. Obviously, we all know that oil stays away from water and vice versa. Well, amino acids span the gamut. Some of them would sit happily in the oil, some of them would sit happily in the water, and some of them are a bit ambivalent. The way that we've color-coded this, the oily amino acids are in blue, the water-loving amino acids are in red, and the ones that are somewhat ambivalent are shown in green. And that was Carl Woese's observation back in the 60s, and he went on to speculate about what it meant. What we did in the PhD, to cut a long story short, was to take these measurements of this particular property of amino acids and ask how non-random is the genetic code. In the manner in which we asked it was suggestive of the Darwinian process, wondering if this was a a very deliberate outcome of evolution. Let me try and unpack that for you. The most common kind of mutation that we know about in molecular biology is the substitution of a single genetic letter. We call that a point mutation. Where it was a G, it becomes an A, or a C, or a T, or any other such single substitution. It's a point mutation. Now, when you think about the genetic code, change one letter of a genetic code word, and that's going to have a variable effect on the intended meaning of a gene that is a function of the genetic code that you're using. The genetic code defines, when you change the letter of one word, what the new meaning is. So what we can actually do with a computer is to measure the difference between the intended amino acid and the one you actually get, according to all the possible point mutations that occur within a genetic code. So, because it's very tedious to do it by hand, you ask a computer to walk one code word at a time, looking at every possible one-letter substitution, looking at the meaning that results, take the difference in hydrophobicity between the new amino acid and the old amino acid, and summarize all of that into a single global average that says that is the susceptibility of this genetic code to point mutations. If you get a big number, it means that on average a point mutation given this genetic code causes a big difference in the amino acids that are being encoded. Small number means you get a very small difference. Why do we think that was important? Is because amino acids aren't as different from one another as, I was about to say, as letters of the English alphabet, but that's the whole analogy. Letters of the English alphabet aren't equally different from one another. If I were to take the words I'm saying to you and replace all the S's with Z's, it would be much easier to understand my intended meaning than if I replaced all the S's with a vowel. Right? That's essentially what we're trying to capture here when we're looking at how susceptible is a genetic code to the average effect of mutations. We believe that hydrophobicity was particularly important because when I tell you that a linear sequence of amino acids will spontaneously scrunch itself up into a three-dimensional shape, the cause of that is the patterns of hydrophobicity in the amino acids. Long story short, although it's imperfectly understood, the oily amino acids find one another just like oil does in a pan of spaghetti, and they find the middle of the protein molecule, exposing the water-loving amino acids to the exterior. That's what reliably gives you a shape from a sequence when it comes to proteins. So if you have a genetic code where random small changes tend to substitute oily amino acids for water-loving amino acids, it seemed intuitive that that would screw up protein making. If instead you can produce a genetic code where even when you go wrong, you substitute one oily amino acid for another oily amino acid, chances are fairly good you can come up with something like the intended product at the other end. Does that make sense? I cut out a lot of slides to see if I can do that in words. Good, 20 years of trying to do that talk, and I can now do it in words. Great, fantastic. Okay, and then of course what was so innovative and wildly crazy that I got away with a whole PhD from it, although people would laugh today, was that nobody had actually thought to use computers much in the mid-1990s. So we simply got a computer to generate a large sample of alternative theoretical genetic codes that frankly could exist, but don't. Measured the average effect of mutations in them, and plotted out to see where nature's code lies on the resulting distribution. And that's the answer. That was my 15 minutes of fame, was a very cheesily named paper called The Genetic Code is 1 in a Million. We published in Journal Molecular Evolution back in 1998. We were doing the work almost exactly 20 years ago, which is odd to think, 5 miles away. But here's the distribution of our theoretical computer-generated codes. Here's the susceptibility to point mutations. If you're at this end, then every time a point mutation occurs, you're substituting very different amino acids, meaning you're getting a horrible outcome. If you're at this end, you have a code in which the average effect of mutation is minimized. And this is the code that nature has been using for 3.5 billion years. Now I've got a lot of people very interested, because prior to that, there was some speculation about the origin of the genetic code, but it was at best tenuous that natural selection had had anything to do with it. We'll wheel in another analogy and talk about the long straight roads you often find in America. And think about telegraph poles as you go down that road. From where you stand, the telegraph poles in the distance look like they're closer together, right? Which is sheer function of limited perspective. That's typically how we have viewed events very early in the evolution of life. And where we stand, they all look like they're bunched up and we don't really know much about them. As our technology and our science progresses and we see ever more clearly into the past, we find out that points that we assumed were all scrunched up together are just as equally separated from one another as points that are much more recent and near to us today. So what I'm saying there is that I think there was a time, probably extending as far as the mid-90s, when we just kind of regarded the origin of life and the origin of the genetic code and the origin of the central dogma as being all one central mystery. We would now see those things as very widely spaced out, with an awful lot of explaining to do in between each one. And this was one piece of evidence that says the genetic code itself appears to be, or at least consistent with, an awful lot of Darwinian evolution in order to come up with an optimal system that minimizes the effects of mutation in a primitive evolving world. Does that make sense? Look for a couple of you. Okay, good. Thank you. Want to emphasize at this point, we were not the first people to do this work. I mean, it goes all the way back to Carl Woeser's observation in the 60s. Plenty of other people who tried to do it. We were just lucky enough to have a computer. Yeah. Okay. A wonderful point towards the end of the PhD where I can literally set it running for 3 days and go off and do something else and just wait for the answer. Sadly now it would take about a second. So, yeah, here we go. Okay. It's carried on for 20 years since then. An awful lot of people have said more intelligent things since. But rather than waste your time on the nuances, let's just say that it is a result you can take to the bank that there is something very, very non-random about the genetic code. That much is pretty universally accepted at this point. No, I'm tempted to say so much more because that's what I researched. I won't. But let's fast forward now to a speaker you're going to get, I think, later this afternoon. Yeah. Simon Conway Morris wrote a profoundly intriguing book, was it 2010, called Life's Solution: Inevitable Humans in a Lonely Universe. Show of hands who's read that or aware of it. Good, yes, of course, that's what brought a lot of you here. I was extremely flattered as a very young scientist to be quoted within that. One of the early chapters dealing with early evolutionary events and the origin of life. He talked about this result in the genetic code, interpreting it to mean that the genetic code, the way we experience it, was pretty much an inevitable outcome. That there was so much Darwinian process at work that indicated to him that this genetic code was likely to occur no matter where or how life had begun. Something like this would end up. And so although this quote wasn't about the genetic code, this of course captures the essence of his book, for those of you who don't know it. His whole book was an argument that far from its myriad of products being fortuitous and accidental, evolution is remarkably predictable. And it was just lovely that early on he chose to find his work in the genetic code and say, look, that's what I'm talking about when it comes to life's earliest evolutionary steps. So that's kind of part one, if I remember my slides well enough. Yeah, okay. That tells you what the central dogma is, what is the finding, and how that was being interpreted. Part two attempts to put that in a context that brings us back to the bigger questions of meaning and faith and science. And this time I want to ask you a question that I literally asked my undergraduate class last week, and I got appallingly ill-thought-out answers. So I want to see if you can do any better. Okay. It's a terrible question, actually, the worst kind of teacher question, so apologies to the teacher in the room. If I were to say to you, complete the sentence in a way that is scientifically valid, A is to B as life is to universe. Think about that. Give me an analogy. Relationship between life and the universe. Give me something that's like that. Maybe I should keep that rhetorical for the time being. You're welcome to challenge me with alternative answers in the Q&A. But that question, in a nutshell, is a very poetic way of expressing the emerging science of astrobiology. That's how I would self-declare these days. I'm an astrobiologist. I work a lot with NASA on the origin of life. NASA is increasingly intrigued to think about the strong chance, we think, that it's happened more than once, and what would be the properties of life elsewhere or wherever it occurs. The key to that question is asking what science can tell us about the relationship between life and the universe. And so, in some ways, what we're all doing as astrobiologists is trying to fill in our own version of A is to B. Some background, if you didn't know this, and what you did so many beautiful lead-ins yesterday, Jeff, I thank you for that. I always tend to see that as the spirit at work when we're doing the Faraday. We didn't talk to each other, I promise, but lovely things said. Yeah, Al Milky Way, you went for 3 times this number, which is a lovely point that physicists are not half as exact as they would like us biologists to believe. Our Milky Way contains somewhere between 100 and 300, maybe even 400,000 million stars. Okay? And we now know that it is one of a similar number of galaxies, each of which contain that number of stars. It's a beautiful image. Look at it. My slides are not proprietary at all, so they will be posted. Look this one up, better yet go to the original, and look at this image of a space telescope pointing towards the center of our galaxy, showing hundreds of thousands of stars crammed into the core of the Milky Way galaxy. Still a minute fraction of the number that are the galaxy, but just some intuitive sense of how many of these things there are. Lots. Okay? Now, if you would ask 20th century scientists, particularly around the 70s, 60s, 70s, but frankly all the way through the 90s, about the relationship between life and the universe, you would have come up with answers that are typically something like these two. There's Jacques Monod, who says, "Man knows at last that he is alone in the universe's unfeeling immensity, out of which he emerged only by chance." Deep emotive language in there that I would challenge you is not scientific language, but at least as far as that scientist was concerned, was the logical way to interpret the science of his time. Alone, unfeeling, chance. It's interesting, this is the time when Albert Camus was writing L'Étranger, when, why am I blanking, but the Catcher in the Rye, all of these existential angst novels about the modern condition. And that's what the scientists think they've discovered. Go up to 1999, and Steve Weinberg, a physicist and cosmologist, says the final laws of nature are utterly impersonal and quite without any special role for life. Slightly more prosaic account, but certainly Steve Weinberg would say that that was absolutely a scientific statement. A vast universe, unimaginable number of stars, and all the laws of science tell us we're a meaningless accident on a planet that's going around one of them. Well, long story short, we can look and ask how that's changed over the 50 years since the 1960s. One way to do that is to look at the Drake equation. Frank Drake guiding a workshop back in '61 about the chance that extraterrestrial intelligent aliens existed. We're going to simplify it today and just look at 5 terms to make a simple point. When he formed this equation in '61, everyone knew that there was something in the region of 100 billion stars in the Milky Way galaxy. So it occurred to him that if we could figure out the fraction of these stars that form planets, the fraction of these planets that are capable of supporting life, the fraction of these habitable planets where life actually emerges, and then the fraction of those where biological evolution takes life towards sentience, we would come up with a number that estimated for us scientifically the number of extraterrestrial intelligent aliens that would be out there. So look and see how those numbers have come along. Within the last 10 years, an absolute revolution has occurred within planetary astronomy. To say that far from thinking that planets are exceptionally rare things, that there is now reckoned to be, on average, slightly more than one planet per star. So let me unpack both ends of that. Every science fiction thing you've ever seen, Star Wars, Star Trek, was based on fantasy, because when it was written, the astronomers, if they'd been asked, said we don't have any reason to believe that there are planets out there. That's part of what was feeding into a Jacques Monod or a Steve Weinberg. But within the last 10 years, we've actually found scientific data that on average, each star contains one or more planets. There's a Nature paper just making that point. This is largely the work of the Kepler telescope, but more broadly, the story of improved instrumentation, which is able to detect these things. I can't resist just giving you one example of what these telescopes are actually doing. I can't do it in England, I have to do it in American terms, but if our sun were a grapefruit sitting in Baltimore, the nearest star, the nearest one to our sun, would be a tangerine sat in Arizona. Okay, I think in European terms it's roughly equivalent to London to Baghdad. Now, our Earth would be a grain of sand about 17 places away from the grapefruit. And a telescope hovering above that grain of sand is able to detect another grain of sand going around the tangerine in Baghdad. That's our instrumentation. That's what we've only just learned how to do. And that's the basis of us saying now that there is an average of slightly more than one planet per star. Quite astonishing stuff. So, we now believe that there are 100 billion stars and about 100 billion planets, at least, and counting. What fraction of planets per star can support life? The short answer is we don't know, but back to our extremophiles. In the last 10 or 20 years, our molecular technology probes have discovered life everywhere. Everywhere. To the extent that we can now reliably identify at least environments in our own backyard of the solar system that could support life as we know it on Earth. That's transforming our view of how many planets could be habitable. We don't know, but we would strongly suspect the answer now is many. And if you're starting out with 100 billion, that's many, right? So what's the fraction of these planets where life evolves, where it actually emerges? Again, we don't know. That's really my active area of research. We've just come hot off the Gordon Research Conference on Life's Origins, one of the big ones that occurs, and I can confidently tell you we don't know. Which is what makes it fascinating, that's why I do it. But what I can tell you are some interesting clues. We now know that there is pretty reliable information that life on our planet actually dates back to about 4 billion years old. 3.5, which was quoted yesterday, is unambiguous microfossils. We're actually seeing the fossils of life. But that's life after a considerable amount of evolution. Using slightly more indirect techniques that, well, I won't bother to explain, but we're growing in confidence we're seeing life down here. Just to put that in context, remember that Earth formed 4.5 billion years ago. It didn't even cool down until about 4.2 billion years ago. And we think that by 4 billion years ago we had life on our planet. That again is new insights, relatively speaking, within the last 2 decades. Growing in confidence, in fact, the real problem here is we're running out of rocks to test. The oldest fragments of rock on our planet that we can find are about, we're finding signs of life in them. So life might well be older. There's a minority view that life may have actually come from Mars in the first place, because Mars cooled down slightly quicker. One of the best reasons for exploring Mars rocks of that age, which we're doing right now thanks to NASA, is that Mars has a much better geological record of that time because it does not have plate tectonics, which recycle rocks. There are actually also more rocks of the right age sitting on the surface of the moon that came from Earth than there are here on Earth. Because every time a meteor whacked into Earth, it threw stuff up. Some of that landed on the moon. Meanwhile, everything on Earth has got recycled through plate tectonics. So we're set to learn a lot more, but really I'm saying that it's almost conservative now to say that life originated down here. We don't know how it happened, but if you ask a slightly different question of when it happened, the best clue we have is that life is not terribly difficult to get started once you've got a habitable environment. And remember, I've just told you, habitable environment means something a lot broader than it used to. Earth back then was nothing like Earth today. There was no oxygen in the atmosphere. In fact, we're now understanding that most of what makes Earth different from the other planets in the solar system is a result of life, not a cause of life. Life has formed this planet, such as putting oxygen into the atmosphere. Interesting quote for you. More than half of the minerals that exist, types of mineral that exist on Earth, are there as a product of life. There aren't that many minerals in the absence of life. That's kind of interesting. Sort of weird thing that comes out of astrobiology. Okay. And I did want to mention to you this plot, which shows you the cosmic abundance of the chemical elements due to very well understood processes that occur on the inside of stars where chemical elements are made. Stars are the ovens which heat up subatomic particles or atoms enough to smush them together and make heavier elements. According to very well understood nuclear synthesis within stars, this is the abundance. Hydrogen is the most abundant element, followed by helium, then oxygen, then carbon, then you get nitrogen, neon, I want to say that's xenon, silicon, sulfur, and down there is phosphorus. Those of you who've done any chemistry will know that things like helium and neon and xenon are known as noble gases because they can't undergo any chemistry. They don't react with anything for reasons that don't concern us. But what that tells you is that life as we know it is made out of the most abundant stuff that can possibly undergo any chemistry whatsoever anyway. So a second clue, life started on our planet extremely early on, and it started out of the most abundant stuff in the cosmos. If life were made out of rare chemical elements, that might be a clue that it's difficult or unusual to get it started. If life started on Earth after 1 billion years of waiting, that might be a clue that it's kind of difficult and you need just the right life conditions. If you find life springing up almost as soon as our planet exists in a cooled down state, and if you find it building itself out of the most abundant chemical elements throughout the cosmos, then those are clues that the origin of life is not difficult, even though we, with our mortal human understanding at this point in our progress, don't understand how it happened. Remember again, 3.5 billion years, life has failed to upgrade or change that. These extremophiles use that. And as far as we know, everywhere in the cosmos is experiencing a constant rain of that onto its planetary surfaces. This is what leads NASA to think we probably aren't that far much alone. Okay. So, can't resist just showing you this addendum to it. That, by the way, that last graph was a logarithmic scale, which we met briefly yesterday. But this is it just shown spatially. Hydrogen is the most abundant element, followed by helium, followed by oxygen, carbon, neon, iron, nitrogen, a slightly different way of measuring the systems. But there you go, taking us down as far as sulfur, reminding you that that's what life's made of. Phosphorus remains a bit of an anomaly, and we can talk about that at length if anyone wants to. But two things to notice. Number one, the most abundant chemical compound in the universe is water. Simply because so all that sci-fi about aliens coming here to steal our water, you might just as well walk to Egypt to find a grain of sand. You could probably find one on Hastings Beach, right? Why would you bother? Okay. Number two is we actually match our cosmos better than we match the surface of our own planet. Our bodies, our chemical constituents, match the cosmic abundances of elements better than they match the surface of our planet. Now, I'm slightly cheating there. We've got a great geologist in the room, but I'll say one word called differentiation, which is responsible for the way that the elements in our planet have settled themselves out. But just at some sort of spiritual / scientific level, think about that. You match the universe better than you match your own planet. You are cosmically entwined. You're made of the most abundant stuff. Joni Mitchell sang it, and Carl Sagan said it, but you're made of stardust. But more than that, you're actually deeply enmeshed in the very chemical abundances of this cosmos. Think about your A and your B right now. And think about your Jack Monod and the meaninglessness and the chance that was being touted as science in the late 20th century versus a view that tells you that life appeared on our planet as soon as it possibly could, is made of the most abundant cosmic chemistry, and that the chemistry of your body matches the universe as a whole better than it even matches what's happened to separate out our planet. In fact, it's not just the chemical elements that are delivered to Earth and to every other planet. We now know that the most fundamental building blocks of life, on the whole, are made through space chemistry, astrochemistry if you want to be proper about it. But we can study things, whether we simulate them in laboratories or study meteorites that fall to Earth, that represent for us the chemistry taking place extraterrestrially. We find the same results. Most of the building blocks of the central dogma are there. I highlight the amino acids because they're the ones I've loved to study for the last 10 years. But you'll also see on there all sorts of other bits and pieces that are the fundamental biochemistry of life, delivered for free by the cosmos to the surface of this planet, probably a big clue as to how life got started so very, very early on. I'm over time, so I just wanted to finish by saying all of this is telling us that to an astrobiologist, an awful lot of stars, an awful lot of planets, many of which are habitable, and life seems to be very easy to get started when you've got the right conditions. Which, for my money, means that probably the most important question astrobiologically moving forwards is, given life, how much of it involves intelligence? I won't use my slides because I've got carried away and talked too much already, but I'll point out to you that this is why Simon Conway Morris thinks it's so important for us to assess whether evolution reliably delivers something like a sentient human out the other end. This is why he would be so interested early on with a genetic code that ends up more or less like ours, no matter how it begins, because Darwinian processes would produce one that minimizes mutation. Now, the place where I disagree with Simon is to think that the debate is solved. We'll see what he says this afternoon. He would claim that our genetic code was an inevitable outcome. I wouldn't. I think that given the pieces of the puzzle, 20 amino acids, 4 nucleotides, code words of length 3, and so on, something like our genetic code seems to be a likely outcome. The problem with that sort of argument is all a skeptic has to do is move us one step back in time, change one detail, and say maybe now everything else changes. You can always use the analogy of endless movies that have explored time travel and the paradoxes it brings about, my favourite being Back to the Future. But the whole point there is no matter what you change, you just move one step back and change something else. Everything in front of it now begins to unravel in a different direction. What if there were 19 amino acids or 21? Amongst the slides I'm not going to show you, for 10 years I've been asking that question. Do our set of amino acids appear to be a particularly good set? Well, once again, we've got a ridiculously strong answer. Yeah, it seems like these 20 make a very, very good set of building blocks, and I can talk about that in Q&A. Which sort of reinforces Simon's point. It's all inevitable. Alien life will be just like us. Well, maybe. But what if what if there were 19 or 20 that were very, very similar? Our result wasn't strong enough to say it'll always be exactly these 20. It says there'll be something like these 20, we think. We then don't know how that will flow forwards through 3.5 billion years of life. We don't know whether there really is inevitability in the evolutionary process. And I don't know how we're going to know that because I don't know how to render that a scientifically testable proposition. What I am going to do is finish you just by saying, there's all stuff we don't really need to talk about that says, 3 challenges that I perceive of finding God or locating God, choosing to locate God within the inevitability of evolution. Number one is that point I've just made. From a secular and scientific perspective, no matter how many repeatable steps we identify in biological evolution, a critic can always say, what if we move one step back, further back and change a detail? Bend a blade of grass, whatever it is. The classic butterfly effect, chaos, whatever you want to call it. How do we get past that problem? No matter what I show you is inevitable, move me one step back, change a detail, and everything else changes in front of it now. Really what we're looking at is the problem of induction in science. Induction is when you claim a pattern or a generality based on seeing something, you see a pattern beginning to emerge. You see it once, you see it twice, you see it 3 times. You're like, oh, it's always going to happen. Now the philosophers and theologians in the room will tell me that Cardinal Hume pointed out back in the 1700s that that's a fundamental problem for science. Our claim to repeatability of experiments rests on the idea that physics tomorrow will be the same as today. Seems like a pretty good guess, but of course we can never know that, right? Sun's always risen this far. But really, this is induction at an even bigger level. No matter what we find that is inevitable, one step back, change a detail, now what? And there'll be an infinite series of questions we would have to ask like that unless we can figure out how to take a fundamentally better approach to testing repeatability of evolution. But then 2 theological points, and I really will finish. Number 1, in your experience and in your spiritual life, is God likely to have left a smoking gun of divine presence within the universe? Or is God more subtle than that? When I think about inevitability of evolution as a clue to theistic evolution, I worry that it sounds a little bit to me like the sort of natural theology of William Paley in the 1800s, but just much more sophisticated, in tune with current science. It implies that God wanted to leave a very heavy clue, so that if you're actually paying attention, you see God. That's not the God that I've met in my life. The God that I've met is one who always allows me to choose from second to second whether I believe or don't. Theological point 2. Just a really genuine, humble, open question to something that Jeff was saying yesterday. When we perceive a God who didn't make things the way we find them, but made the laws that would naturally produce them the way we find them. What was your phrase for that, Jeff? Can you remind me? God who makes the mechanisms, or
Things make themselves.
Yeah.
making things make themselves.
Actually, Charles Kingsley, not Jeff Hardy.
Well, okay, I mean, a small difference, but okay, thank you, right. Thank you, that was Charles Kingsley.
Much better facial hair.
Frankly, sir, that's a choice, you can work on it. But no, anyway, more than once in the history of Western civilization, the history of science, the idea of finding a God who makes things that make themselves has led people to a deism, a remote God, a prime mover, a God who created the laws of physics such that everything else would take care of itself, which leaves a very open question about whether and how we find God at work in our personal lives day to day today. Now, of course, I know Jeff believes in a theistic God who's hands-on today. I do. I'm guessing an awful lot of you do. I just wonder whether looking for inevitability in evolution is yet another way that some of us could accidentally fall into a more deistic view of God. Well, sentience was bound to occur through evolution. God created evolution, therefore God created us. Where does that leave us in our day-to-day relationship with God? I don't claim to be a theologian. I don't claim to know. I thank you ever so much for your patience, but let me stop there. Thank you.
It's over to all of you to ask your questions. Joe, could you? If you could use the microphone, it helps people at the back.
Hello, thank you very much for your talk, which was fascinating. My question is about the 5 basic elements early on the periodic table. Is it possible that you could replace one of them, say carbon? I know this sounds a bit science fiction.
No.
Because we've heard about carbon-based life and so on.
Yeah.
Could you replace that by one other and run some simulations and see if something similar or different develops?
Computers are almost at the point where they can do that. It is not a silly question. That's the sort of thing that NASA very seriously is challenging scientists to get creative and think how to do. I would suggest that at the moment, the favoured answer is it's possible, but why would you when carbon is more abundant than anything else? We know that carbon is reacting with those other 5 through space chemistry in meteorites and then getting deposited to the surface of planets, and the carbon is good enough for making life as we know it. So most astrobiologists would say there may well be weird life out there. That's what we call it, because we're very scientific, weird life. Yeah. Might be weird life out there, but we're much more likely to find oddly mundane life because of the way and the abundances with which things are made. Computers are just about getting to that stage. I see a bright future for that sort of question, if you're feeling like it. You know, seriously, anyone can have a crack at this, so yeah. Thank you.
A question here?
Can I say thank you so much for an absolutely brilliant talk.
Oh, thank you.
I'm just, just in reply to that last question, aren't they doing that sort of thing with combining the silicon? Isn't that the whole point of the silicon thing?
Well, silicon has long been talked about, and it would be the best substitute for carbon. At Earth temperatures and pressures, it tends to spontaneously combust very easily, which isn't very helpful for building life. But, of course, there may be colder conditions where it would be more stable. And the cost of working with carbon would be so heavy that you'd prefer silicon. So that's the sort of question we do ask.
Okay. Can I ask?
Oh, yeah,
you-
please.
sorry,
Yeah.
it was really to recap on your own slide for your own research, just so I can understand.
Oh, yeah,
it.
yeah.
You showed the slide of all the possible outcomes of all the combinations of the four codes. And did I read it rightly that you said that all but one were worse, but there was one that was better? Is that what you were saying?
Yes, but just to tiny detail, we only sampled 1 million. And due to the weird math involved, there are countless billions. So it's a ratio of 1 in a million would be better, according to those calcs.
Okay, so my question is, well, I mean, immediately I saw that, I thought, have I got it right? So everything would be worse apart from one. So what is that one, and why?
Wonderful question.
What?
Sorry, yeah, do you want to...
What's the one and why?
So,
This is on a postcard too.
yeah, no, what is the one? Remember, it's 1 per million, and there are billions. So there are actually countless millions that are better. They're just incredibly rare. So that's why I don't want to focus too much on the details of the one. But the better answer to your question is to say that this is the interesting point we reach when you're trying to do inevitability and evolution. How well do we measure what we set out to measure? If there's any degree of error or oversimplification using a single measure of hydrophobicity, there's some error bars around the one. Could be that life and evolution just got very good, but didn't get perfect. And there's good reasons to think that's the way that evolution works. Could be we didn't measure it perfectly. And the more you chase this down, the harder it is to discriminate those two.
What would it look like if it were on the perfect one? What would life look like?
We don't know. Because we'd have to do such computer simulations to imagine what that would then lead to. Yeah. Thank you. Those are great questions, all of these.
Wouldn't you want there to be a little probability of change being brought in answer to that? Because that's the very way we try and
Innovation,
niches and innovate things. Yeah, so if you had something that was so absolutely stable, you just have a Volkswagen Beetle for the rest of your life, for the rest of the universe.
Yeah.
You know, whereas now we have Mercedes and we have Toyota Corollas and all sorts of other things.
Absolutely. And that would be a good way to answer it, is it's actually optimized for the process, not for the perfect outcome. It's optimized to leave you a little bit of wriggle room that mutations have an effect. Now, there are hours worth of lectures on that computer, at least, where we go into this in nauseating detail to suggest this curious paradox of evolution. The more you've got something that works, the more you want to hang on to it. So I can quite imagine, and it would make perfect sense, that very, very early on, life was actually more in favour, or systems had an advantage if they mutated readily. The more they accumulated useful genetic information, the more they would want a genetic code that buffered them against further deleterious damage, while giving them that wriggle room.
What you're saying is the perfect process is better than the perfect outcome.
That's much better than I was saying it, thank you. Yeah.
That's quotable right there.
Isn't it? Isn't it? Just grow some facial hair if you wouldn't mind. Yeah.
Great, thank you. I think there's a question over there at the far end of the row, and then we'll come over on the other side.
Yes, thank you for your talk. You talked about DNA sequences being linear with a beginning and an end.
Yeah.
Can you get loop DNA with interesting properties, in particular for longevity of life?
You can get, yes, so a bacterial chromosome technically is one big loop. When I say linear, it's more this sense that it can be read in a straight line. It's not branching, it's not two-dimensional, it's one-dimensional. So bacteria very cleverly encode to us the concept of a start and an end on an infinite loop. Because the starting point is the key to knowing where to find genes and how to read them. So although, yes, you can end up with loop DNA, you can end up with all sorts of interesting stuff, functionally think of it as a straight line because that's how biology uses it. But thank you for that correction. I appreciate it.
And then there was a question over here.
Thank you. My question is about the Drake equation. I mean, it's a fascinating thought to think through how probable is it to find life on other planets. On the other hand, without having a mechanism how life arises, I'm always suspicious about his statistical argument. For example, the factor planets that support life is actually the wrong term, I would say. There must be fraction of planets that on which it is possible that life arises from scratch. That would be interesting.
Yeah.
Because once life is there is no doubt that it can adapt to different environments and so on. But I think this factor is wrong. And so I ask the question, is it not a lot about fundraising, this great equation for the NASA?
Thank you, excellent question. Funnily enough, no. In fact, NASA will never use the Drake equation because we regard every term in it as flawed. Those of you who studied the process of science will realise that it is not that science finds answers to the questions it was asking. Science learns it was asking the wrong questions. Everything in the Drake equation since 61 we now know is pretty much the wrong question. Habitability being a great example. We now know that life changes planets more than planets set themselves up for life. We now know that moons and asteroids are as likely to be possible sites for an origin of life as planets are. But we could go through every term, again, hours of lectures I won't bore you with. So you're absolutely right. But they don't use it for fundraising. And in fact, Drake really used it not as an equation to solve, but as a way to lead a workshop like this one for scientists in 1961, one of the early attempts to bring together all of the natural sciences to say, what do we know? And I think in that capacity it still functions as a guide to a discussion. But that discussion could very fruitfully take on board the sort of criticism you've just raised. Yeah. I use it because it's the cheapest, quickest way to get through a point. But thank you for that. And yeah, you're absolutely right. There's holes we can poke in every bit of it. I still find it useful as a didactic exercise in the classroom. We did it last week. Spring I teach astrobiology and I love it. That's what I'm rushing home to. Got the students to solve it. And then to recognise that not one of them had been able to come close to an N of 1. N of 1 means there is one example of life in the universe. Sentient, if you do the whole thing. And the lowest example we had was 50,000. And that was somebody who was utterly pessimistic and out of tune with every bit of science that we're about to throw at them. And that, I think, is still a useful point, even though we could poke holes in every term. So thank you for that.
I would have two questions. First, I would like to hear your take on the, I was actually expecting you mention the left and right amino acids. Right. So if you can please talk a little bit about that. And the second one, being a scientist that is involved in astrobiology as yourself, I would like to know your take on the origin of life problem in terms of the, well, what came first, RNA world and DNA protein world, and what's your personal take on that?
Thank you very much for two questions. Again, answered on a postcard, right? We had a wonderful conference where for a week we were asking these questions and ended up more confused than at the beginning. Second one first, because that's freshest on my mind. My take on the origin of life, I think the concept of an RNA world has reached its prime and is now deteriorating. For those of you who don't know, those who've asked what came first in the central dogma, the ascendant theory since the 90s has been RNA, strangely, that middle player that you've never heard of. Reasons we don't need to go into. But that placed all the emphasis on whether we could come up with a chemistry that produces RNA in the absence of living systems. Best way to produce RNA is give me a living system and it produces RNA. So many millions have been thrown at that. So much effort has gone into it. It's a purely subjective opinion, but I think it's telling us something. That for every inch we get forwards in torturing a system to give us something a bit like RNA, we're getting ever more wildly creative. I've long suspected that may be the wrong answer. I don't know what the right answer is, but I think this telegraph pole analogy works for me. The more we see into the past, the more we're realising that things we just assumed were bunched up together aren't. I think there were many, many precursors to the central dogma as we know it. And I'm trying to think how on earth we can get clues as to what they may have been. That was what we spent a lot of the week talking about. And there's more and more interest on things that we just don't see anywhere in life today, but we can account for that would have properties that could then be upgraded to the central dogma. That would be my take on the origin of life. Amongst that, this issue of chirality, for anyone who doesn't know but cares, carbon can make 4 bonds. I'm not going to try and do it with fingers. It can make 4 bonds. Long story short, that leads to a curious property that for any given molecule that is bonded to a carbon atom, there are 2 mirror images that can be made. That if you make them, they're not the same. They're functionally equivalent, but the arrangement of atoms is such that you cannot rotate this one so that it becomes the other one. Yeah, we use hands and wave, we literally hand wave, but yeah. So that's known as the chiral problem, and life is very specific in its chirality. So all the sugars that life uses are one chirality. All the amino acids that life uses are another chirality, and so on. So one of the questions long-standing in the origin of life is why? Why those chiralities? Again, personal opinion, but thank you for at least asking me to, I'll be clear, this is a personal opinion. I'm convinced that chirality was an outcome of evolution, not a prereq for life. I think of it in an analogy of the Industrial Revolution, that life learned to standardize its bits as an efficiency mechanism early on. And one piece of standardization is just an arbitrary agreement. I'm going to use left-handed amino acids, right-handed sugars. Because using standard pieces leads to efficiency, as we saw in the Industrial Revolution. You go back before the Industrial Revolution, there was no such thing as standardized screw threads. Well, that meant that you always have to check that the exact same craftsman had made your thread when something, there's efficiency to just agree standardized units. I think that's a very deep point that I choose to see exemplified in the Origin of Life community as a growing concern. That so much of what we take for granted, we used to try and draw a straight line between chaos and life as we find it, shortest possible path. I think we're now imagining a more and more wiggly path, where life as we find it today is a fundamental upgrade, and that's giving us new insights into how it could have begun. Very personal opinion, so please take that with a grain of salt. Okay.
Okay, last question. Sorry.
My question may be a bit simple for this audience, but it strikes me that do you think God actually intended us ever to know the origin of life? In terms of, at the moment we can look at a test tube, we can put the key elements into it, but we can't create life.
Right.
Is that a question that do you think we'll ever solve that question, or do you think God intended that we were just to accept that?
I mean, I think your question is an excellent question, particularly for a Faraday workshop. My personal opinion is I don't see that God did not intend for us to understand it, based purely on the ridiculous things we've been able to understand about our universe. I find it astonishing that we can talk confidently about the birth and death of stars that are in other galaxies, while we still struggle to understand or predict how a jury will behave. That tells me that there's something odd here, that God intends us to understand the universe if we choose to. I don't know quite what to make of that. It could well be that God doesn't intend us to, and that eventually we'll realise we're never going to understand it. But I don't see anything in the science that I've witnessed in my lifetime that tells me that God has drawn clear lines saying you are not to know this. Just based on what we have found out, that blows my mind. I don't know why God wants us to know it. I mean, back to that lovely quote yesterday, the sheer arrogance of informing God about what we should and shouldn't know. But I mean, if I were in charge, I'd be like, God, couldn't we understand each other and do relationships better? And I'll give up the stars. But that's not the way God intended it, right? So, yeah. So thank you for a wonderful question.
Profound and perplexing
Right.
note. I think we should say thank you so much.